Did you say you're not posting until Monday, the
I
lecture?
said I said probably I can get it up by Monday, the rest of this lecture. The reasoning is I have to record all of my structure and function lectures tomorrow, so I can't probably record these as well. So I'll get up the rest of these by Monday. Awesome. All right. Hopefully that will be okay for y'all. All right. So looking at these different T cell components of T cell differentiation. So this is looking at the movement from T cells through the bone marrow, the thymus, the lymph node, and peripheral blood. So this is really getting into the comment that I think was brought up before about what are the T helper sites, Th1, Th2, et cetera, and what are they doing? So from our bone marrow, our T cells are going to be maturing in the thymus, and our T cell precursor comes from the bone marrow, and then our T cell in the thymus cortex has both CD4 and CD8 components on its membrane. Then in the thymus medulla, it becomes specific for either a CD8 T cell or a CD4 T cell, depending on which component it maintains, it upregulates, and which component it eliminates. So we call this like double positive and single positive. So initially, all T cells are double positive, and then they leave one or the other. From there, our CD8 naive T cells will eventually mature and become cytotoxic T cells in the lymph node. Our CD4 T cells will eventually become our helper T cells in the lymph node. And these helper T cells are going to go on to be involved in cytokine release in the lymph node as well as in the blood. So let's look at our helper T cells because those are the more particular types. So there's 4 different helper T cell subtypes that we can find in the peripheral blood. Our Th1 is going to be really upregulated by either interferon gamma or interleukin 12, but interleukin 12 is the main player that's going to upregulate Th1 cells to be produced or differentiated. They're downregulated by these things here. So that's kind of what this slide is showing, is the upregulation and the downregulation. From there, our Th1 cells will produce both of these cytokines, but primarily interferon gamma is the primary actor that it's producing to activate macrophages and activate our cytotoxic T cells. And this response, we can typically call this more of a bacterial response or an infectious response, but we also, in fact, could say this is an intracellular response. This is going to target things that can target stuff that are inside of cells. By activating macrophages, we're encouraging opsonization and phagocytosis. By activating cytotoxic T cells, we're encouraging porphyrin granzyme destruction of our cells that have been tagged for destruction. So Th1, while an infectious response, we often associate with a response to something happening intracellularly. Now, our Th2 helper T cells, it's going to be primarily upregulated to differentiate into a Th2 cell by IL-4, interleukin 4. And then its main products that it's producing is interleukin 4, again, so it has a positive feedback mechanism, 5 and 10. So we'll see that these are the main cytokines that's going to produce. They're going to stimulate its release. These 3 cytokines, their job is to do things that are more extracellular, allergic, or dealing with parasites. So we'll see that it activates eosinophils, which are our big parasite helminth fighters. And it'll also increase IgE, our immunoglobulin that's associated with allergies. So Th2 kind of tends to get the name of our allergic response, but you could also call this the extracellular response to some type of antigenic episode or event. You'll then see Th17. Th17 is induced to be produced by IL-23 as the primary actor, but these other ones will influence it as well. And its main production is going to be interleukin 17 and 22. And it's going to be actually an inducer of inflammation and inducing neutrophil activation. So it's a big inflammatory responder, our Th17, giving this kind of inflammatory response. And then counter that with Treg. Treg, it is a helper T cell, but it's our regulatory T cell, meaning that it's going to prevent autoimmunity. So it's going to tamper down an immune response. You can see some of the cytokines produced that will stimulate Treg production, And then it's going to secrete tumor or tissue growth factor beta, interleukin 10, and interleukin 35. And its goal is to tamper down our immune response and prevent autoimmunity. And we can see here that Treg producing IL-10 is what inhibits Th1 to be produced. So if we had a big kind of infectious inflammatory response and we're trying to kill off a bunch of cells that have stuff inside of it, and the body's like, excuse me, you're actually killing our own cells, creating an autoimmune response, it will upregulate Treg, upregulate IL-10, and then downregulate Th1. So you can see these interrelationships between these different cell types. You can also see that Th1 produces interferon gamma, which inhibits Th2, and Th2 produces IL-10 and 4, which inhibits Th1. So that's why you can sometimes say, as an ND, we can talk about, well, if we want to downregulate an allergic response, let's try to upregulate our infectious response or our Th1 response in the body. Or let's try to promote Treg to kind of downregulate them all. So you can see kind of these different ways that we've learned to manipulate the immune system based off of these helper T cells and their cytokines that they secrete and these different feedback loops. We look at it here. This is exactly kind of the same picture, just showing it in a different way and kind of talking about some of the different functions that I already mentioned. So we can see intracellular pathogens, stimulation of IgG, organ-specific autoimmune diseases, defense against helminths, allergy, and then defense against extracellular bacteria or fungi, autoimmunity, and chronic inflammation. Remember, this is our big inflammatory responder. So it's just causing inflammation across the board. So it's good for things that are outside of cells as well. So these are our T helper cell differentiation pathways.
And it's the Dr. Pacho, it's the
Yeah.
Th17 response that if that gets dysregulated, then that's what is linked to autoimmunity, or because Treg kind of balances it.
Treg balances it. If this goes out of whack, this can send us down an autoimmune pathway, but so can Th1. So, I mean, realistically, any of them can, but because of the heightened inflammatory response that Th17 has across the body, that's why we tend to associate it with being potentially leading us down an autoimmune pathway.
Okay, gotcha. Thank you.
Yep. Yeah. Absolutely. And chronic inflammatory pathway because of the amount of inflammation it can produce. And having that activation of neutrophils, that high activation of neutrophils, that's a big inflammatory response in itself. And that can take a while to tamper down. Yeah. Excellent. Awesome. If you wanted to see a picture of just, again, this is exactly same. It's just showing here the memory cells that are produced at the same vein once these cells become activated. So they were naive, then they got exposed to antigen, then they divided. Some of them are going to become the effector cells, meaning that they're going to have their actions. Some of them will be memory cells, so they'll remember the antigen for next time. Showing some of the molecules involved in T cell activation. This is where you get all these different like proteins and receptors and C stuff, T stuff, and it gets really like muddled in our brains, right? So some that we already know. CD4, this is identifying it as a T helper cell. So we have this CD4 molecule here, this signal transduction molecule here. TCR is just T cell receptor. This is the thing that's going to bind with that antigen, whatever that is. And it's the thing that's going to interact with our MHC class 2 for this instance, because we're dealing with the CD4 T cell. When we look at our CD3s or CD28s, these are signal transducers. So they can essentially help send signals as well, like CD4. We just don't talk about them as much because CD4 is really the one that we get to name the cell from. These are more helpful if you're studying immunology, but not as helpful from just overview cell perspective. And then this is actually an adhesion molecule that helps to bind and helps to the cell stay stable when it's interacting with its antigen presenting cell. So I'd say of these things, which ones should you know? Know CD4, understand TCR, T cell receptor, and understand your MHC classes. Those would be the three that are the most important things to understand and to be able to identify on your T cells. cytokines or co-stimulators. So when we look at our macrophage lymphocyte interaction, then this is the words that kind of go along with this last picture. So our Th1 cells can secrete interferon gamma. We saw that with our Th1 cells on that big slide where I had the 4 different categories. Our Th1 cells secrete that interferon gamma cytokine, or essentially a chemokine. This will increase the ability of monocytes and macrophages to kill off microbes, and it's enhanced by the interaction of T cells with specific CD molecules on macrophages. So T cells, besides secreting this, they'll also directly interact with macrophages via CD molecules, not necessarily CD4, but with different CD molecules to upregulate, upstimulate that response. And then macrophages will activate lymphocytes via antigen presentation in reverse. Macrophages can present antigens back via their MHC complexes. Macrophages are one of the antigen presenting cells that has an MHC class 2, so it can directly interact and communicate with our CD4 helper T cells. And so this is showing here, this is, again, this effector functions of our Th1 cell, macrophage activation. So we see our antigen presenting cell, either dendritic or macrophage, has its ingested microbes in those phagosomes. It's presenting antigens via MHC class 2s. It's secreting IL-12. It's stimulating our naive CD4 T cell to become an effector Th1 cell that secretes interferon gamma, saying the macrophage should continue doing its job. This non-activated macrophage should start killing off microbes, and this is the microbe that it should kill off. And so this is going to upregulate our macrophages activation. What we don't see here is the direct binding that will occur between this macrophage and the CD4, the toll-like receptor on our naive CD4 T cell. So there will be direct binding besides this chemical stimulation as well. And this is, you can see kind of your classically activated or alternately activated macrophage. And your classically activated macrophage, you can see monocyte becoming activated here versus monocyte becoming activated here. Monocyte being a macrophage before it goes into tissue. Macrophage being a monocyte once it's moved into the tissues. And you can see microbial toll-like receptor ligands, interferon gamma, that's what's upregulating this process. IL-13 and IL-4 is what upregulates this process. And then we can kind of see the different actions based off of what's being asked of the macrophage. So if we have that like really infectious response, we'll see reactive oxygen species, citric oxide, lysosomal enzymes, things that are going to really help with phagocytosis. And then we'll also see chemokines that are pro-inflammatory being secreted. Versus if we have this alternative pathway activated, we'll see IL-10, tissue growth factor beta, which is more anti-inflammatory effects, so that will potentially inhibit the inflammation. And we'll see our proline polyamines and more wound repair and fibrosis, because macrophages can do both. So depending on the needs of the body, they'll either be activated classically to do this job, which is what we've been talking about, or they can be activated alternatively to help muck up the system and calm down an immune response. And it's all based off of the cytokine signals. That's all like what it's being communicated to, what signals are being released is what it's going to essentially follow as a guide and to what type of cell it will activate into. So I think these slides are really helpful, just looking through, I'd say probably this whole section. This slide, and not this one. This slide and this slide, just to kind of understand these different activation pathways that are present. Okay, our cytotoxic T cells aren't as exciting. I mean, they're exciting in that they can literally apoptose other cells and kill virus-infected cells, tumor-infected cells, but they do so in the same way as natural killer cells. They release those cytotoxic granules, and they are essentially, they get through these little porphyrin pores, and then they're going to digest and break down that cell from within, and then the cell apoptoses and dies. And you see these apoptotic bodies. The cytotoxic T cells do have a CD8, which binds to MHC1 on virus-infected cells. That's how they're signaled to release their granzymes and their perforins to know that this is the cell that's targeted for them to kill. And our regulatory T cells, I already kind of mentioned this before. This is just words saying what was on that other slide. They're helping to kind of balance out our immunity. T cell activation, again, walking through kind of the same process here. If you just need to see it in words, how T cells are activated, essentially antigen presenting cells present antigen to them. T cells then bind with those antigen presenting cells via MHC2 molecules and CD4 receptors. And then from there, those T cells are going to become activated. They're not naive, they're specified now. And then they, depending on the activation needed, they will mature and they will start producing their cytokines based off of the antigen that was identified. Okay. This is the good stuff now. Now we're going to get into cytokines and function. So this cytokines, I want you to categorize them not as cell-mediated immunity, but humoral immunity. And you'll see them acting in both the innate and the adaptive immune system. But we'll look at some cytokines first that are really highly active in our innate immune system. So this little image here is just showing our natural killer cell, macrophage, and then inflammation in general in a blood vessel. And we can see these are neutrophils traveling down. And then when the microbe is getting kind of reactivated, who's sending what signals where? So when our macrophages and natural killer cells get activated by a microbe, our macrophages are sending a signal of IL-12 out, which will stimulate natural killer cells. Natural killer cells are sending an interferon gamma message out, which will activate macrophages. So you have this like cycle continuing. Macrophages also will send out TNF and interleukin-1 as well as chemokines. Now let's go to the blood, not necessarily locally. And that's going to help neutrophils identify where to come to and where to move out of the blood vessel via diapedesis or extravasation into the tissue surrounding. And so these are really your inflammatory response in this innate immune response. So again, in words, macrophages and natural killer cell activation is IL-12 and interferon gamma. Inflammation induction is tumor necrosis factor, IL-1 and chemokines. Our antiviral effects is going to be your interferon type 1, your interferon alpha, and interferon beta. These are going to be your primary antiviral effects. And then differentiation of T cell subpopulations is going to be IL-12. So these are kind of some important components to recognize in your initial innate immune response. Now, when we look closer, we can look at these tables and see specifically macrophages can secrete a lot, right? We've seen here, they already are secreting like 4 different things. So we can see kind of the cytokines that macrophages secrete, the actions they have in the body, and maybe if there's a pathology associated, what that pathology is. So essentially, IL-8 is the chemokine, the chemotactic factor for neutrophils, and neutrophils are recruited by IL-8 to clear an infection. So that's kind of what they're used for in a pathologic situation. So I encourage you to really focus on, to start with, understanding these cytokines here. So IL-1, 12, interferon gamma, and tumor necrosis factor. If that feels good, then you can go and start digging into some of these tables, identify like this is who's secreting what, macrophages. These are the things they're secreting and the actions they're having in the body, and potentially a complication to those actions. Some of them good, some of them maybe not so good, right? This is facilitating a granuloma formation, which could be helpful and not helpful at the same time. So I would encourage you to start to look through these. I'm not going to read through every single one, but I tried to categorize them by who is the major secretor and then what are their major actions. But I think summary slides like recognizing IL-1 and tumor necrosis factor are your big inflammatory responders. That's huge. I think that's a big place to start. Our T cells, depending on the T cell type, we know they're going to secrete different stuff, right? So in general, T cells are secreting, when they secrete IL-2 or IL-3, we can see it's going to self-stimulate growth of either helper, cytotoxic, regulatory T cells, and natural killer cells, or supports growth and differentiation of bone marrow stem cells. So any T cell, if they're secreting these, they're trying to essentially make more T cells. They're trying to upregulate, increase proliferation, increase differentiation, increase cell division because they recognize they need more T cells present. So that's how I'd look at IL-2 and 3, because we're not looking at a specific T cell type. We're looking at T cells in general. Now, Th1 and Th2 cells, they're going to secrete some different things. So Th1, we have that inflammatory response. We see interferon gamma being one of the big things that it's going to secrete. And so Th1 is going to have that inflammatory response, secreting interferon gamma, really helpful for killing off things inside of cells, stimulating stuff to get rid of stuff that's inside of a cell. Th2, we can see all these other cytokines that are going to be secreted. And we'll see that our Th2 response, we're kind of dealing with some B cells. IL-4 is encouraging more helper Th2 cells, and it's encouraging B cells and some antibody class switching to get us Ig from getting IgE and IgG antibodies, probably from IgM. IL-5 is helping B cells differentiate again and helps class switch to get IgA antibodies. So we see both of these are actually stimulating B cells. They're secreted by our Th2 T cells. And we can kind of go on and so forth, but we start to kind of get a feel for the difference between Th1 and Th2. Th1 being more pro-inflammatory, try to destroy what's inside of cells that are affected. Th2 is more extracellular, trying to encourage B cells to help out with antibodies, because we're probably looking at an allergic response or some type of parasitic or helminth response. And that's outside of a cell. So we need more help to be able to destroy them. The interferons as a category or a class, I want you to think about them as part of our innate host defense and really part of our viral infectious response. They interfere with both RNA and DNA viruses, and they can decrease or downregulate our infected cells to resist viral replication. And they also are upregulated to show other cells that they've been infected. So anytime you're thinking, if you have a question about what would be your cytokine involved in a viral response or response to a virus, think your interferons, alpha, beta, and gamma. They're going to be your main viral responders that you'll find in your immune system. And this, I like this. This is just a simplified table showing innate immunity, some of the cytokines, the players who secrete them, their targets and effects. Adaptive immunity, the cytokines involved, who's secreted by, and their targets and effects. So if you wanted to focus on one slide, this could be a helpful table that you could focus on for your self-study for your cytokines. Okay. All right. Not hearing any major rumbles, so I'm going to move on with antibodies. So with antibody structure and function, I've been mentioning the Fc regions this whole time. So the Fc region of our antibody is this kind of base region down here. So you see Fc here. The Fc region is essentially constant. It has a carboxy terminal. It does complement binding, and it really confers the isotype of the antibody. Versus the FAB fragment, the fragment antigen binding, is the part with the light chain here and the heavy chain here. And this is where our antigen binding sites are. So this is the portion, the FAB portion, that is going to bind to the specific antigen, versus the FC portion is the stable portion that determines the type of isotype of the antibody. The heavy chain you'll see is part of both the FC region down here and the FAB region, but the light chain is only participating in antigenic binding. That's the light chain's job. So when we talk about our immunoglobulin types or isotypes, there's 5 total. We start at IgM or IgD. IgD exists, but there's not as much known about its functionality. And then you'll seroconvert from either IgM or IgD in this order, in order of IgH. So you'll either start with IgM and then go AGE via seroconversions, or you could start at IgM and then go IgD, AGE. But it'll always be in this order, AG and E. That's your order of seroconversion. You'll never go backward. You'll never skip one. It'll always be in this direction. So what do I mean by isotype switching? We can see this is a B cell via helper T cells and cytokines. They are starting to produce their different subclasses of antibodies. So IgM is the one that's first, and its main function is to help activate complement, which is part of our immune system, our innate immune system. We'll see IgG will be produced if we have a lot of interferon gamma, that strong inflammatory response, that viral response, that infectious response, that Th1 response. So IgG will be produced, and this is going to encourage phagocytosis, complement activation, essentially intracellular tagging for opsonization for destruction. Our IgE is going to be stimulated by our IL-4 or our Th2 cell response, right? They secrete IL-4. IgE is our helminths and our allergens, right? Our type 1 hypersensitivity reaction, creating that mast cell degranulation. That's where you see your IgE. And then IgA, there's a lot of cytokines involved here. Pretty much anyone that's secreted in a mucosal tissue would be involved in this IgA secretion. And this will be mucosal immunity, so transport of the IgA through the epithelia. So we can kind of see these different subclasses here, their functionality, and who's being triggered by what. So looking at them individually, IgD, like I said, is found on the surface cells of B cells and in serum, but we're unsure about their function, but it does exist. IgM is produced as the primary response to an antigen. So it's the first on the scene. It helps to fix complement, to kind of start that complement activation process. It also has an antigen receptor on the surface of B cells, and it has multiple binding sites. I don't know if I have a picture of it. I'm bummed. I don't have a picture. Well, it's here. It has multiple binding sites. You can see like all these different binding sites around it versus the other ones are much smaller. But these multiple binding sites bind to antigen while the humoral response is starting to pick up. So it's really does a great job at picking up a lot of antigen to start sharing it around with the other cells of the body to upregulate this immune response. IgG is the main antibody in the secondary response to an antigen. It's the most abundant antibody type in our serum. So at any given time, IgG antibodies would be the greatest amount, regardless of antigen type, right? This is also going to fix complement, so bind to complement. It'll opsonize bacteria, neutralizes bacterial toxins, as well as viruses. And it is the only isotype that crosses the placenta, and so it can provide passive immunity for infants. Our IgA antibody will prevent attachment of bacteria and viruses to mucous membranes. It does not interact with complement. It's the only one thus far that does not interact with complement. It'll cross through epithelial cells, and it's produced by our Peyer's patches in our gut. It's really helpful against gut infection then. It's also released in secretions, tears, saliva, mucus, and breast milk. And if we had to choose one that's the most produced antibody overall, this would be IgA, but it's less populated in the serum because it's in the mucosa. So it's the most produced, but it's not as populated in the serum. IgG is more populated in the serum than IgA. And then IgE antibodies bind to mast cells and basophils. They are able to cross-link with allergens when they're exposed to allergens, and they mediate the type 1 hypersensitivity response because they're able to essentially stimulate the release of inflammatory mediators when they are bound to their antigen and they're bound to a mast cell. So that will be that histamine response, that degranulation response. They also can contribute to immunity to parasites by activating eosinophils and stimulating their response, which their main job is to fight off our parasitic infection. Those are our antibody classes. Okay. All right. So then complement. Complement people confuse, but I'm going to try to keep it as simple as I can. We could make it a lot more complex, but we're going to try to just focus on the main pieces of complement. Complement is a bunch of plasma proteins. There's a bunch of them. They all have names that start with C and then have some numbers and have some letters. They're all synthesized in the liver, and they're all involved in innate immunity and inflammation. One specific part of complement is the membrane attack complex, or the MAC complex, or MAC attack. And MAC defends against gram-negative bacteria. And we'll actually see it has an ability to break apart bacteria cell walls. So it's an important component of complement. Complement has three ways that it can be activated. It can be activated in the classic way by antibodies, either IgG or IgM, by those, remember I said they can fix complement. That's the classic way of them activating complement to start this innate process. It also can have an alternative pathway of activation, which is directly interacting with microbe surface molecules as a part of the innate immune system. And it can have the lectin complement activation pathway, which involves mannose or sugars on the microbial surface, glycoproteins that it's interacting with. So we'll look at some of these different pathways. So we can see that all three of these pathways are essentially activating complement. And the first step of complement, the early steps onward, and we'll look at these steps broken down here more and more, is this protein, C3 or complement 3. When we have activation from any of these pathways, it's going to get converted into C3b because we essentially broke off this tiny piece called C3a. And C3a is involved in upping our inflammatory response. So think of C3a as causing more inflammation. From there, we'll see C3b will go on. It'll be deposited directly on the microbe, and it will opsonize that microbe and be able to stimulate it for phagocytosis. So this is how we're able to identify capsular bacteria, encapsulated bacteria, is via complement C3b opsonization, ability to coat the microbe with these proteins, and then a macrophage can identify it and phagocytose it. From there, our late steps, we get involved with C5, complement 5, and complement 5 will start to kind of break down into these components. And 5A, just like 3A, is an inflammation promoter. But these 5B portions are going to essentially combine together, all these proteins combine together to create this complement 9, which is going to be our MAC attack, our membrane attack complex, which will be able to lyse our microbe. So all of this from these tiny little proteins produced by the liver, and all part of the innate immune system. So we don't need any specificity of activation. We just have to know that something is harming us. And then complement will become activated via one of these three pathways. So if we look at this in words, in the classical pathway, we'll see the early steps involve complement proteins 1, 4, and 2, and late steps involve all these other complement proteins. Our early steps for lecithin, relatively similar. We just have one change here. And then alternative, we have kind of a whole different pathway here. And then you can see that all the late steps, all of these complement proteins are the same. So late steps don't change. It's the early steps that might alter based off of the activation pathway. Now you're like, Dr. Petra, what do I have to memorize this? No, I would focus on understanding these are the three activation pathways. This is what they mean. And then in general, understanding kind of what's this process of complement activation and what does that mean for innate immunity. So looking a little bit closer at some of these complement proteins, just to have some definitions again. So our opsonins that do opsonization is our 3B, 4B, and IgG. And so remember, when we looked at this picture here, 3B, we don't see 4B in this picture, but 3B doing opsonization is what I'm referring to when I'm talking about opsonins here. C3b can even bind to lipopolysaccharides. Anytime we opsonize a bacteria, we're increasing our body's ability to phagocytose it. Now, our cytolysis is literally our breaking down our bacteria. We can lyse the bacteria, and it's using C5b through 9. So all those proteins, 5, 6, 7, 8, all the way through 9, will form a pore essentially called the MAC, the membrane attack complex. And that can lyse cells, and it does a really good job actually of neutralizing Neisseria species. So our Neisseria meningitidis and gonorrhoeae. anaphylaxis or stimulation of inflammation. I would just think about this as pro-inflammatory. All the things that have A's are pro-inflammatory or pro-autoimmune. So it's C3A, 4A, and 5A. All are inflammatory molecules. And then neutrophil chemotaxis, 5A also will essentially signal neutrophils to come to this region or this area. So this is what our opsonization looks like. We already kind of saw this, but this is our C3b binding to the microbe, opsonizing it. Our macrophage then can recognize it now. It's like, oh, yep, this was before it was encapsulated. I couldn't see it, but now I can. And then it's able to phagocytose and kill the microbe. Our MAC complex, so C3b binds to the microbe, and then it activates components of complement. You'll then get stimulated later down the road, the MAC formation, the membrane attack complex here, and then it'll lyse the microbe. You essentially are creating a pore through the cell membrane, and then via osmosis, the inside of the contents of the bacteria and the outside contents of the cell or of essentially the host intermingle, and the bacteria cell wall can't maintain its osmolarity anymore. And so the cell literally breaks down. Our inflammatory responses, all the things with A's, they are going to send out an inflammatory response, essentially activating mast cells and neutrophils. And so those are going to upregulate inflammation further. And so those are your big pieces of your complement. So it doesn't have to be that confusing. It can be if you really want to get into the protein weeds of it all, but I would stick to this level of knowledge should give you enough to get through your NPLEX. More would be more of a graduate level immunology class for an immunologist. Okay. Okay. All right. Let me see where we're at. Okay, this is good. We're actually doing pretty good. So we're almost done actually with this portion. So the last piece here is just respiratory oxidative bursts, kind of biochem. We had some of this in our biochem lecture, but just a moment for rapid release of oxygen species, ROSs. We talked about all the ways that bacteria neutralize this, trying to use catalase and peroxidase and all those things. But our respiratory burst is the activation of our phagocyte NADPH oxidase complex. So we can see this NADPH being activated to NADP. Our oxygen gets essentially an electron, so it's becoming more stimulated. Then we see conversion with superoxide dismutase, and we'll see that will activate kind of this cascade process here. But you'll see hydrogen peroxide being created. If bacteria have catalase, they will break down hydrogen peroxide into water and oxygen, and they will therefore not undergo this respiratory burst, this destruction by reactive oxygen species. If they don't, though, then we can take this essentially hydrogen peroxide, and then with our enzymes, we can actually convert it and add things like chloride on there and other items which can then be used to attack our bacteria and break them up. So this is that conversation here. So superoxide dismutase then is going to activate our catalase glutathione peroxidase, our glutathione reductase, and our glucose 6-phosphate dehydrogenase. So kind of this cascade of enzymes being activated. And I mentioned that catalase positive organisms can neutralize this portion here. But the kicker is if we can keep our reactive oxygen species from getting neutralized, and then we use myeloperoxidase, this is going to be what sputum gets its greenish color. We can add on something like a chloride ion, and then this is what we can use to essentially neutralize bacteria. And this plus mucus and the broken down parts of the bacteria is what we see in our green mucus as we start to fight off a disease. And this is in a phagolysosome. So inside of a phagocytic cell, once the phagocyte has bound or combined with its lysosome, we get a phagolysosome. And so that's what's occurring inside here to try to break down and destroy this bacteria. And if the bacteria has catalase, like our Staph aureus or Aspergillus or any other catalase positive, they can withstand this process and avoid getting broken down via this mechanism. There's others that we can use. The kicker, though, is we see this biochem connection with the need for our NADPH. So that's like the connection with our pentose phosphate pathway and why we need those stores to be adequate. And that's also where we get the connection with glutathione, because that's in the pentose phosphate pathway. So that's kind of how we have these connections between those pathways, is in our like bacterial fighter system, is directly using that NADPH in this process to create the ROS to then be able to kill off our bacteria. The last little piece I have before I'll stop and just open to questions for the end is just a couple components on mechanisms of defense versus a virus specifically, and then I also have versus a helminth. In our innate immune system, we'll see that there'll be inhibition of the infection and induction of an antiviral state via type 1 interferons. I mentioned that interferon alpha and interferon beta, as well as killing of our infected cells via our natural killer cells. On the adaptive immune side for viruses, our B cells and antibodies will do neutralization and opsonization to essentially neutralize the viral components and opsonize cells that have viruses in them to tag them for phagocytosis. Cell-mediated CD8 is actually going to be killing off infected cells, and CD4 is activating both CD8 T cells as well as B cells to create more antibodies. So that's our innate and adaptive response to viruses. And we can kind of see it here. This is just showing exactly what I said already, just innate immunity versus adaptive immunity. And if you're like into little flow charts like me, you can see virus infected cell, type 1 interferon, so either alpha or beta, the uninfected cell versus an infected cell. You can see protection from infection versus being targeted to be destroyed. So kind of these two pathways. This is our process to destroy our infected cells by natural killer cells. We already talked about this earlier, about what they do with their porphyrin and their granzyme. And neutralization of viruses without antibodies or with antibodies, allowing for binding of the microbe and the infection of the cell to target it for destruction. Antibodies also can have a protective effect on cells. This is showing essentially target cell, infected target cell. So kind of these two pieces here. We can neutralize antibodies, can block the uptake of our microbes into the cell receptors. So we can kind of use antibodies to also help neutralize to avoid uptake of viruses inside of a cell. And then our CD8 killer cells. So again, going through all these processes again that we've already looked at, just specifically looking at viruses. Now, parasites, I did want to spend a moment on for innate immune systems, so protozoa and helminths. They're mainly resistant to the innate immune defenses, but we do still try to use them. And this is complement as well as phagocytosis for protozoa and eosinophils and macrophages for helminths. Then our adaptive immune defense, you can kind of see, I tried to put some specific components of like who might be where. But B cells, for protozoa, B cells, CD4, Th1 cells, and CD8 cells are involved because these tend to be intracellular. Antibodies, the B cells tend to target more entamoeba or plasmodium. Interferon gamma production and stimulation of macrophages as leishmaniasis. And cytotoxicity, again, plasmodium, so that intracellular piece. Versus our helminths, we're looking at more of a Th2 response. So we're trying to stimulate IgE production, eosinophils, and mast cell degranulation to break down those parasites that are located outside of a cell in the extracellular space. And then this is just showing you those processes, just so you can see that degranulation and eosinophil activation. And then same here. It's just showing it at a closer view of eosinophils doing their job. All right. And that's where we'll stop here because we're pretty much to the point of vaccination, which I think is a good like start point. The rest of this is honestly not that much. It's just a little bit about vaccines, and it's a lot of practice questions. And then I have a little bit of immune pathology, and then some like kind of overarching tables. So I encourage you to look through the questions on your own. When I record, I'm probably not going to like go through the questions that slowly. So I encourage you to go through the questions yourself as well to do some practice on this section. Okay, you made it through microimmuno with all of your brain knowledge and brain candy we have now, an exploding brain. What questions, support, guidance can I provide you?
This isn't about our stuff today, but I'm wondering if you're able to upload more anatomy practice questions for your exams. And then I think you said you did upload other micro and immuno ones, right? Okay.
Yes. Yeah, microimmuno, I already have like 3 or 4 or 5 up there. Anatomy, I didn't do a great job at finding more challenging anatomy questions. That was my struggle. A lot of the questions I was finding were at the same level or simplistic. I'll try to still do some digging to see what I can find, but I was unsuccessful at finding more challenging anatomy questions to kind of further push your study. So that's why I haven't posted anything there yet. But I will still do some digging. I'll see what I can come up with and unearth. Right. Anything else? How far out are we now?
Like 2.5 weeks.
Okay. So we'll have one more weekend next weekend. That will be kind of a grab bag of physiology, pathology, a little genetics, heredity smattered in there. I am in person. I hate lecturing online like this. So my goal is to be in person, and I believe we will be there as long as there's no more new snowstorms. So that's the plan next weekend. Friday, we'll have a true practice test in person. So please come if you want that experience. It'll probably be your last opportunity to have an actual like proctored practice exam with pencil and Scantron before your test. It'll be physiology and pathology focused. And then that Saturday, we'll run through lecture. I think we have from 9 to 4 that Saturday, and we'll be in 107. I'm going to save a good hour at the end for questions. So I want to make sure I give some time for whatever has come up. And then at that point, you'll have essentially a week and a half of self-study. Your last weekend, we don't meet because I think your test is on the 6th of February, if I'm correct. Yeah. So the last weekend before, we don't meet, so you can just do whatever you need to do. But obviously, I'm a resource if you need anything, moral support, whatever. Okay. All right. Any other questions or things I can support you with today before we sign off and eat some dinner?
Thank you.
Okay. Of course, I'm thinking of all of you. You're in my thoughts. I'm sending you all the good energy, positive vibes through the interwebs and the universe. And come find me if you need anything on campus during the week. I'm assuming we'll be back Monday, so I'll probably see y'all around. Thanks.
Thank you so much.
Bye. Take care.
Thank you.